Hello everyone, my name is Kai Cao, and I'm a PhD student at the Chinese Academy of Sciences. It's a great honor to have this opportunity to present our work, Unsupervised Topological Alignment for Single-Cell Multi-Omics Integration. In this talk, I first gave an introduction to the background and challenges of single-cell multi-omics data integration. Then, I will give a literature review on the related work. After that, I will introduce our method UnionCom, which is an unsupervised topological alignment for single-cell multi-omics data integration. Then, I will give some experimental results, and UnionCom outperforms other methods on both simulated and real-world datasets. Finally, I will give a summary and conclusion. Recently, the advent of single-cell sequencing provides high-resolution omics profiles at cellular level, and it offers us the opportunity to unveil molecular mechanics of fundamental biological problems. And single-cell multi-omics data such as gene expression, DNA methylation, and chromatin accessibility are obtained from the same sample or tissue. In these multi-omics datasets, cells are usually heterogeneous, and features are usually high-dimensional. So from a computational perspective, we can obtain multiple high-dimensional data matrices in different forms. So why data integration is important? As mentioned before, different omics datasets are in different high-dimensional feature spaces, and joint analysis of this data is difficult. So it is important to find a data integration method for downstream analysis. The integration will build connections across modalities or find correspondences information between cells. Besides, it will also provide a more comprehensive molecular multiview of intrinsic biological processes. However, the integration of single-cell multi-omics data poses fresh computational challenges. First, due to technical limitations, the cells in different datasets are usually unpaired, which means there is no cell-to-cell correspondence between datasets. Second, these multi-omics datasets usually have unmatched features, so they are in different feature spaces. Third, there may be nonlinear geometrical distortions within dataset. For example, the two datasets have similar intrinsic low-dimensional structure, but they are in different feature spaces and have different geometric distortions. Our job is to find the complex nonlinear map between datasets. Since the data integration is very computational challenging, this area is drawing heavy attention from both machine learning and the data science community. Many single-cell data integration methods have been developed for the batch effect correction of single-cell datasets in one modality. One of the pioneer work is MNN, which finds mutual nearest neighbors in a common feature space, and it uses this information to align datasets from one modality for batch correction. However, MNN has two assumptions that batch effect is almost orthogonal to the biological subspace, and the batch effect variation is much smaller than biological effect variation between different cell types. The assumptions are too restrictive for single-cell multi-omics data integration. And some latest methods, such as SRAT and Harmony, try to project data into a common subspace. SRAT uses canonical correction analysis, and Harmony uses principal components analysis to find the subspace. Then, they align multi-omics datasets in the subspace. These methods are mainly designed for batch correction and have a cluster assumption, which does not work well on data with intrinsic nonlinear continuous structures. Most of the previous work depends on a common feature space and linear transformation. However, MMDMA, the most advanced nonlinear method for multi-omics data integration, does not require correspondence information and can deal with unmatched features. MMDMA is based on a kernel method, and it measures the distance between distributions of the datasets in a shared latent space by maximum mean discrepancy, but it assumes the same low-dimensional structure between datasets. MMDMA is close to our method UnionCom that I'm going to introduce. Our method UnionCom is a distance-based method, and it works on the general assumption that cells from different datasets shall dominate intrinsic low-dimensional structure. UnionCom is totally unsupervised and needs no correspondences between cells, and can deal with unmatched features. UnionCom is a nonlinear topological alignment method, and it has ability to identify data-specific cell types. As shown in this picture, these black points are specific cells in dataset Y, the dominant structure including red, green, and blue points. UnionCom is set on the foundation of Guma algorithm. Suppose we have two datasets X and Y, and assume that Y has a dataset-specific structure. We first embed each single-cell dataset into a distance matrix. We use the two distance matrices as new embeddings of dataset X and Y. Then let's call this matrix Kx and this matrix Ky. Our goal is to find a matching matrix F to minimize such an objective function, where F subject to these constraints. This integer constraint limits the algorithm to one-to-one correspondence. This inequality constraint gives the algorithm ability to identify specific cells. F acting on Y is equivalent to find a match between dataset X and Y. So minimizing the norm of subtraction of two matrices is equivalent to find the optimal match. In fact, it is close to a classic graph matching problem and also a quadratic assignment problem. But our UnionCom allows Kx and Ky to have different dimensions. To better deal with the nonlinear topological structure, we propose to compute a matrix of the shortest distance paths based on a k-nearest neighbor graph within each dataset instead of Euclidean distance. It has been proved that the shortest distance path of the kn graph converges to geodesic distance on the embedded nonlinear manifold. Then UnionCom uses the optimization problem described before to match cells between datasets. Here we make two improvements of Guma. First, to deal with the nonlinearity and the distortions of the datasets more flexible, we introduce a global scaling factor alpha. Second, because the original integer optimization problem is NP-hard under an exhaustive search strategy and difficult to optimize, we relax the integer matrix to a right stochastic matrix. Then the constraints will be a convex set and the algorithm will be more computationally efficient. Therefore, this change relaxes one-to-one match to probabilistic match between cells. Afterwards, to optimize F and alpha, we develop an efficient prime dual algorithm. It is based on the augmented Lagrangian function. This is the objective function, and the mu and the lambda are dual variables for equality and inequality, respectively. This is the augmented term that is designed for stability of algorithm and the acceleration of convergence rate. After getting the matching matrix F, how to project multiple datasets into a common space is still a challenge. Here we propose a solution that topological structures are preserved well and cells with the same types are aligned together. To preserve the topological structure, we adopt 2KL divergence, which proposed by Tisney. To align the two datasets better, we introduce this coupling term. This term aligns matched cells by using matching matrix F so that the matched cells are close in the common space. After this step, UnionCom gets the result of integration of datasets. In order to evaluate the performance of the integration, we use label transfer accuracy, which is widely used in domain adaptation or transfer learning community. High score of label transfer accuracy means a better performance in integration. Next, we come to the experimental part. Here we first simulate a complex tree structure, and then add Gaussian noise to form dataset X. For dataset Y, apart from Gaussian noise, we rotate and transpose the data. Then we randomly map the dataset X and Y into 1000 and 500 dimensional space, respectively, so the two datasets are in distinct feature space. We use UnionCom to integrate the data. The result is shown here. The colors in left panel picture represent different datasets, and in the right panel represent different cell branches. The result shows that UnionCom integrates complex tree structure well. And UnionCom outperforms other methods, such as MMD, MA, SRAD, and Harmony. UnionCom puts the data into the common space better and has a highest label transfer accuracy. MMD, MA has the second highest label transfer accuracy, but SRAD and Harmony perform poorly because they are linear methods and not designed for topological alignment, and cannot deal with complex tree structure well. Furthermore, we test whether UnionCom can identify dataset-specific cells. We simulate a trafficate tree structure, and we remove the green branch in dataset X, so dataset Y has specific cells in green branch. We use the same random mapping and integrate them by UnionCom. The result shows that UnionCom successfully identifies the green branch and aligns shared dominant structures well. UnionCom also outperforms other methods in this simulated data and has the highest label transfer accuracy. In this case, MMDMA preserves global structure well, but it assumes that datasets share the same low-dimensional structure. It cannot work well when datasets have specific structures and achieves relatively low label transfer accuracy. Harmony has a high accuracy to match these branches, but it cannot preserve the global tree structure. Here we also test UnionCom without global scaling factor alpha, and the accuracy drops in this situation, which verifies the effectiveness of the factor for nonlinearity and distortions. Next, we test UnionCom on real-world single-cell multi-omics dataset. We first integrate widely used multi-omics datasets, SC-GEM datasets, including gene expression and DNA methylation. The datasets share a linear structure with 5 types of cells. UnionCom aligns the same cell type between gene expression and DNA methylation quite well in the common embedded space by locating samples between datasets on a common region with similar distributions. UnionCom also has a highest label transfer accuracy in this dataset. MMDMA has a relatively low accuracy, which is below 30% in the default parameter setting. But we also found that in their latest paper, it can also achieve 60%. SRAD locates cells from the gene expression dataset in an ethereal region surrounded by cells from the DNA methylation outside, and Harmony has similar situation with SRAD. UnionCom can also integrate more than 3 omics datasets. Here we give an example of integrating 3 modalities, including gene expression, DNA methylation, and chromatin accessibility in SCNMT datasets. UnionCom successfully integrates 3 modalities into a common space and preserves dominant linear structures of cell lineage quite well. Meanwhile, UnionCom can also be used for batch correction tasks. We use HSC datasets here and found UnionCom has very similar performance with other state-of-the-art methods designed for batch correction. We also test the robustness of UnionCom in Turing parameters, and we found that our method is robust to a widely range of parameter choices. In fact, we use the same parameters in both simulated and real-world dataset integration. Therefore, in most practical applications, you don't need to worry about the parameter tuning and can use the default parameters. At last, I will give a summary and conclusion. Our computational algorithm UnionCom is a distance-based method which deals with unpaired cells and unmatched features. It is a nonlinear tool which aligns complex topological structure efficiently. To align different datasets, we propose a new solution that puts matched cells close in the common space and preserves the topological structure simultaneously. Besides, UnionCom accommodates samples with state-specific cell types. The experimental result shows that UnionCom outperforms other methods in both simulated and real-world datasets. Although UnionCom is computationally efficient compared with GUMA algorithm, we can still improve its computational speed and convergence rate by software optimization and combining with advanced methods such as Adam algorithm. Besides, when datasets are very large, memory consumption is still a problem for distance-based or kernel-based methods. Therefore, we plan to use some technical tools such as geometrical sketching to select a small subset of data for integration. Our software is available on GitHub and Python package index. Welcome to use, and if you have any questions, please feel free to feedback. This is the end of my work. I want to thank to my co-author, Xiangqi Bai, Dr. Yiguang Hong, and Dr. Lin Wan. Thanks for your attention. I'm ready to take questions.